Hello ladies and gentlemen, my name is Walter Kamtiko and I'm going to present on my project topic titled Cumulative Neurotoxicity of Gadolinium-based contrast agents for magnetic resonance imaging in rats with intact blood-brain barrier. Magnetic resonance imaging is an important diagnostic tool that is used in investigation of several disorders, including neurological disorders. In evaluation of these disorders, particularly neurological disorders, gadolinium-based contrast agents, which, for example, include Magnevist, are administered or injected to the patient before the MRI procedure, and their function is to aid in further characterization of tissues. In other words, these compounds, they help or assist the physician in enhancing natural contrast of tissues so that he may be able to quickly identify and differentiate between normal and abnormal tissue. And over the past years, these compounds have been considered safe to the body because they were considered to be swiftly cleared by the kidneys with a half-life of at least 30 minutes to 2 hours. But however, several studies have emerged. These studies proved that upon repeated exposure to this drug, it tends to accumulate in several body areas, including the brain. In the brain, most accumulation occurs in the globus pallidus, which is part of the basal ganglia, as well as the dentate nucleus, which is part of the deep nuclei of the cerebellum. And both these areas are involved in motor coordination. And several studies also have been carried out to assess if this accumulation might have some consequences or some detrimental consequences. But however, as a human study has proved impossible, because people who are subject to continuous exposure to these gadolinium-based contrast agents are people who already have neurological conditions, and the effects of this accumulation might quite overlap with the effects of the already prevailing neurological conditions. And this brings us to the conclusion that an animal model might be most appropriate to use in assessing if this accumulation has some cumulative toxic effects. And this brings us to our research question. Does repeated administration of gadolinium-based compounds produce cumulative neurotoxicity in rats with intact blood-brain barrier? So I think intact blood-brain barrier because people with neurological disorders, let's say multiple sclerosis, those new disorders might be associated with a disrupted blood-brain barrier, which would allow easy access of the drug into the brain parenchyma. So our main objective now is to assess the cumulative neurotoxic effects of this drug on motor function, since it accumulates in areas involved in motor function. And our secondary objective was to assess if this accumulation has some effects on neurobehavioral function. So the research was carried out at University of Zimbabwe Physiology Lab or Physiology Department, and 20 male sprague-doli rats were used from ages 8 to 12 weeks. They were used and they were allocated to 4 treatment groups. The first group was called the control, and it received normal saline. And the other 3 groups, 2, 3, and 4, received 0.05, 0.08, and 0.1 millimoles per kilogram body weight. That is the dose of the drug. And these were kept in a standard room with standard room temperature and with natural daylight, that is a 12-hour daylight cycle. And they were provided standard rodent food. They were also provided tap water, and they were provided wood shavings as bedding. And the experiments were carried out between 8 a.m. and 4 p.m. The drug itself, the different concentrations of the drug, were prepared by serial dilution of the actual drug concentration and to make different concentrations, and alongside with normal saline. And the drug was administered through the lateral tail vein, so it was administered intravenously, and the animals were injected 3 times a week over a period of 4 weeks or 28 days. And on day 0, day 14, and day 28, several tests were carried out to assess motor function as well as neurobehavior. So to assess motor function, we use what is called rotarod test. In rotarod test, the animal was placed on a rotating rod, and the latency to fall from that rod was noted, or the time to fall. As well as also we used suspended wire, and the time it took for the rat to fall from that wire was noted, and it was recorded as the latency. And also, in order to assess neurobehavioral function, we used a method called neurobehavioral functional observation battery, in which several scoring criteria regarding autonomic functions, neuromuscular functions, sensory motor, as well as activity observations. So they were allocated different scores according to the behavior that was indicated on each score. And on the final day, that is the day 28, all the animals that survived would be killed, and the killing procedure involved weighing first, and then chloroform asphyxiation. After chloroform asphyxiation, the animals were then dissected and using guillotine method, and their brains were harvested and put in 10% formalin, and the bodies were incinerated at the Faculty of Veterinary Science. And after all this was done, the data that we would have accumulated, so we already entered the data in Excel, and we used several statistical methods to calculate and analyze our data. We used a Kruskal-Wallis application, a Kruskal-Wallis method to calculate. It was a non-parametric test, and we also used ANOVA, and we used GraphPad Prism to generate our graphs. And from this analysis, we discovered or observed that the latency to fall from the rotarod and suspended wire was significantly reduced compared with control. And so this was motor coordination using rotarod, and this was using suspended wire. So we can actually see that the latency to fall was significantly reduced in both tests, starting even from day 14, and then moving on to day 28. And from the functional observation battery results regarding activity observations, few differences were noted, and only 3 significant differences were noted. One was from activity observations, that is ease of removal as well as arousal. So there were 2 from activity observations and 1 from sensory motor observation, which was in touch response there. But however, in autonomic observation as well as neuromuscular observation, no significant differences were noted. And so this data suggests that based from the main aim of this study, we were assessing the cumulative neurotoxicity of gadolinium-based compounds on on motor function or on the central nervous system as a whole. So this was the first such study that we carried out, that was carried out as far as we know, actually looking into the neurophysiologic effects of gadolinium-based contrast agents. And from our results, our findings suggest that gadolinium-based contrast agents affect motor function, or actually affect function in the central nervous system. And from literature review, this is consistent with the finding that gadolinium, upon accumulating in the brain, it might release the free gadolinium ion, which is an ionic radius which is similar to calcium, and these may compete. So it actually inhibits calcium physiology, that is transport as well as physiologic functions. And in this experiment, the frequency of drug administration would play a role, because the frequency compared with humans, the frequency was quite high. And the finding that autonomic nervous system, there were no differences, was expected, because the drug has not been seen to accumulate in areas concerned with autonomic functions. And in conclusion, this experiment has proven or given us some evidence of gadolinium accumulation. And so this might send out a message to radiologists to be aware of these effects, since it's the first such study to assess the effects. And also, since gadolinium-based contrast agents only affect motor function, we can safely conclude that this data is quite relevant because of its accumulation in those areas as well. And however, this experiment lacked evidence of the actual molecular mechanism that is involved in causing this function. So we would recommend several other tests to be carried out, for example, to assess histopathology to see if this effect on function is reversible or irreversible due to the damage that would have been caused to the neurons. And also, a human study must need to be carried out because this was an animal study. So another human study might need to be carried out to maybe prove these findings that we had. And these are the references that I used.